Now we're going to talk about the autonomic neuroeffector junction. The autonomic neuroeffector junction. Okay, so the autonomic neuroeffector junction is the synapse between efferent and effector organs in the autonomic nervous system. What does that mean? What does efferent and effector organs in the autonomic nervous system mean? What are efferent fibers? What does efferent mean? Or efferent? I feel like I always say that word. What does efferent mean?
Coming from the central nervous system to the organ.
Good. So a signal coming from the central nervous system down to the effector organ. So when we say efferent, we're really talking about that postganglionic neuron, which is bringing some sort of signal or instruction or communication from the brain out to the effector tissue, which is an autonomic nerve, okay, the postganglionic nerve. So the area between a postganglionic neuron and your effector tissue is the neuroeffector junction. So let's just recap. Let's go back a few slides. So we can see a clearer picture of where this is. All right, so let's take this example. We spoke about our two neurons, preganglionic, postganglionic. This second neuron, this postganglionic neuron, synapses onto some type of effector tissue. The area or the synapse between these two structures is what we're really looking at here. This is called the neuroeffector junction. All right, it's going to hold true for the parasympathetic division as well. So the area where a postganglionic neuron communicates with your effector organ. What are some examples of those tissues? What are some examples of those effector organs that we're talking about here?
Smooth muscle.
Smooth muscle.
Glands.
Glandular tissue.
Adipose tissue.
Adipose tissue.
There's one more.
One more. Cardiac muscle, right? So the area where that postganglionic neuron communicates with one of those four effector tissues is called the neuroeffector junction. It's going to be important in the release of neurotransmitter, right? Now the neurotransmitter can differ here. It can be acetylcholine if we're looking at the parasympathetic system, or norepinephrine and epinephrine if we're looking at the sympathetic system. Now, in terms of the release of the neurotransmitter here, they're stored in swellings that we call varicosities. And the release of these neurotransmitters is in response to an action potential being fired in that second neuron. All right, so we get our signal from the spinal cord. It travels out the preganglionic neuron. It meets at the synapse. It travels down the second neuron, which is this neuron, the postganglionic neuron. It gets to a varicosity, and when the action potential gets to this varicosity, it's going to trigger the release of that neurotransmitter, which will cross the neuroeffector junction. And as a result, the neuroeffector junction is not a traditional synapse, okay? It's very different. It's not one neuron talking to another neuron. But what we have are the several axon terminals of this postganglionic neuron coming in contact with this effector tissue, whatever it is. Let's say it's a piece of cardiac muscle. So all of these axon terminals have some swellings on them, and then they come in contact with that effector tissue. Right? Now this image here shows a larger view of a single varicosity. So these swellings on the axon terminal are called varicosities. And if we looked a little bit closer at a single varicosity, we would see that there are some synaptic vesicles within this swelling. And within those vesicles are the neurotransmitter. Now again, it can either be acetylcholine or norepinephrine, depending on where this postganglionic neuron is. So if it's in the sympathetic side, the vesicles are going to store norepinephrine. If this is a parasympathetic postganglionic neuron, the vesicles are going to store acetylcholine. Okay? Now, as we know from previous knowledge, studied along the membrane of this axon are going to be your voltage-gated sodium channels, your voltage-gated potassium channels. What are those responsible for? What are those voltage-gated sodium and potassium channels responsible for along the length of the axon? Good, the transmission of the action potential down that axon. So as we mentioned before, there should be a review. We had myelin along the length of the axon, and in between those areas of myelin, we had a concentration of these voltage-gated channels, which allowed the action potential to jump along the length of the axon. When it gets to the swelling, what channels are present on the swelling itself? What channels result in exocytosis of neurotransmitter? Calcium channels, perfect. So at the varicosity, just like what we saw at the axon terminal, there are the presence of voltage-gated calcium channels along this swelling, along the varicosity. Okay?
This picture,
Mm-hmm.
kind of confusing because it actually looks like it's myelinated. I mean, that's what I think of, like, it's in the brain. Up on the top, that top picture.
Which one? This one?
Yeah.
No, so this is the neuron.
Okay.
It's not really showing myelin.
No, down below.
Here,
Right there, yeah,
this is the axon terminal, but it's different from a traditional synapse because we just have that neuron and those terminals ending in a bunch of swellings as opposed to meeting the way we saw with the synapse, right? Does that make sense?
Yeah.
So when we look at the synapse, let's say at the sympathetic chain, let's say at the ganglia, where we have the preganglionic neuron and the postganglionic neuron. Let's jump back. So let's take this area. Let's take this area. So this is a traditional synapse. Okay, we have one neuron speaking to another neuron. It's like a one-on-one conversation here. This is going to be true for all of these ganglia, as well as the adrenal medulla. When we look at the neuroeffector junction, which is between the effector tissue and the postganglionic neuron, it's not a traditional synapse. We don't have a single neuron talking to that tissue. We have that neuron ending with a bunch of swellings, and those swellings will release neurotransmitter, and it will diffuse across to that effector tissue. Okay? Let's move back here. And that's another thing that we're going to point out is that the neuroeffector junction has two main differences between, or two main differences from a traditional synapse. First of all, they're the presence of varicosities as opposed to axon terminals. And there are many varicosities at one neuroeffector junction as opposed to a single axon terminal at a synapse. The second difference is that it's a further distance. So because we don't have that intimate connection, one neuron speaking to another neuron, those are going to be very closely adhered. Here we have a further distance. The synaptic cleft is going to be bigger because we just have these swellings that are releasing neurotransmitter, and it's like a free-for-all. They have to find their way to that tissue somehow. Okay, so that's a greater distance across that synaptic cleft. So let's talk about the events at the neuroeffector junction. Let's talk about the sequence of events here. And I didn't bring my drawing stuff. Okay, so first thing we have is the action potential arriving at this varicosity. Okay. When it arrives here, it's going to trigger these voltage-gated calcium channels. All right, so remember we said on the membrane of the varicosity are voltage-gated calcium channels. The action potential gets to this varicosity, triggers these voltage-gated calcium channels, and opens them such that calcium rushes into this varicosity, and that will trigger exocytosis. Okay. Now, exocytosis occurs, this neurotransmitter is released into the neuroeffector junction here. And depending on what this neurotransmitter is, if it's acetylcholine, as in this first image, it's going to go and bind to this muscarinic cholinergic receptor. All right? Why is this not a nicotinic cholinergic receptor? Where are the nicotinic cholinergic receptors? Where are the nicotinic cholinergic receptors? Good. So this is at the synapse where we have that one-on-one conversation between a preganglionic neuron and a postganglionic neuron. So on the membrane of that postganglionic neuron, there is where we would see your nicotinic receptors. On the effector tissue, right, is where we have muscarinic cholinergic receptors. So this acetylcholine binds to this muscarinic receptor, and as you can see, it's coupled to a G protein, which will signal some amplifier enzyme downstream, which will bring about your cellular response. Okay, we're not going to look in detail at the cascade of muscarinic receptors. Okay. Let's look at the scenario for a sympathetic postganglionic neuron. So if this was on the sympathetic side, the neurotransmitter is obviously different. It's norepinephrine or epinephrine. It's going to have the same sequence of events. The action potential gets to this varicosity. We open up voltage-gated calcium channels. Calcium rushes into the varicosity. The rush of calcium triggers exocytosis of this vesicle, dumping that neurotransmitter into the synaptic cleft. That neurotransmitter crosses the synaptic cleft and binds onto this adrenergic receptor. Now, bear in mind, depending on where this tissue is, the adrenergic receptor can be different. It can be an alpha 1, it can be an alpha 2, it can be a beta 1, a beta 2, a beta 3. If we say, for example, that this effector cell was a cardiac myocyte, what type of receptor would this adrenergic receptor be? If this effector tissue, this cell, were a cardiac myocyte, cardiac muscle, what type of receptor would that adrenergic receptor be? A beta 1 receptor, perfect. If this were in the lungs, what type of receptor would that be? If the cell were a lung cell.
Beta 2.
Beta 2, good, and so on and so forth. If it were adipose tissue, it would be a beta 3 receptor, so on and so forth. So these two images really help depict what takes place between that postganglionic neuron, the second neuron, and the effector tissue. And we really want to bear in mind that the effector tissue can be different locations, different receptors, different effects.